Cinti, and today I'd like to talk to you about is RNA processing. It's what occurs to RNA after it's been transcribed, and it's a fascinating process. It involves removing some non-coding regions, and it also involves adding a little something to the front of messenger RNA, a little something to the end of it. And so, what's fascinating is that we shall begin. And let's see if we can do that. Let's go over here and click there. Okay, so first off, there's a little bit of a difference between eukaryotic cells and prokaryotic cells. One of the things about prokaryotic cells is that they don't have a nucleus and they don't have introns. And so I'm going to show you a little video later, but basically they don't have to modify their RNA and edit it like eukaryotes. So let's go into eukaryotic modification first. And so there's enzymes in the nucleus that contribute to this modification. And so what happens is, whoops, sorry about that. It's a little bit of a bugger here. So what happens is that in the 5', which is the beginning of the messenger RNA, let me go ahead and draw that. The 5' end of what's called the sort of the pre-messenger RNA molecule, or the pre-transcript. It's modified because there's a guanine. It's one of the nitrogenous bases, and a guanine is added to the 5' side of messenger RNA, and it has a little methyl attached to it, not drawn to scale. It's actually kind of small. And it's like, what's the point of putting that 5' cap, if you will, onto the molecule? And it's thought that the main function is that it helps it to attach to the ribosome, because remember, this is leaving the nucleus, and it's going to meet up with a small ribosomal subunit, and it's thought that it functions as an attachment area to the ribosome. It also helps to prevent the molecule from being diced up a little too quickly by hydrolytic enzymes or RNases in the cytoplasm. The funny thing about that is, ultimately, that's not necessarily a bad thing, because we remember that RNA, we want it to be temporary, but we don't want it to be too temporary. So that's the point there. And so this 5' cap is attached, and so you can see here's a really detailed close-up, and here's a nucleotide, here's the ribose, here's the guanine, and here's the methyl group. So this is at the 5' end of the molecule, so 5' cap. A little animation of this. Here's the 5' side here. This is RNA polymerase, and then here is the cap that's being placed on the RNA. And so here you can see here, not drawn to scale, because of course it's ridiculously large. Here's the gene in here, the start codon, stop codon, and then here's the termination signal, which helps to stop transcription. And then over here is the poly-A tail, and this is the second thing that happens to messenger RNA when it's modified before it leaves the nucleus, is that at the 3', or the tail end of the messenger RNA, there's an enzyme that comes along and attaches a really long, sort of repetitive adenine to this. It's like AAAAA. It's called a poly-A tail, and it can vary in length, but it's a couple of hundred of adenines in length. And what's the point of this? Same as the cap, it's thought to prevent a quick hydrolysis of messenger RNA, and it's thought to help in some ways facilitate attachment to the ribosome, so similar to the cap. And then this is kind of odd, but it's believed in some groups believe that this poly-A tail actually helps it leave the nuclear pore, and I find that to be kind of mysterious, because it doesn't seem like anything longer would have an easier time leaving the nuclear pore, but apparently that is the case. You can see it here as an animation. Here's the 3' or the poly-A tail of messenger RNA. Now, what has to be done next is, if you remember, if I were to animate this, let's go here, let's go yellow. You might think we're done. We have the 5' cap over here, and here's the poly-A tail that's located here at the 3'. But do you remember from our previous discussions that we said that the gene itself has intermittently regions of non-coding segments, and these areas right here need to be edited before the RNA is able to leave the nucleus. These non-coding areas are called introns. And so, introns. And so what we want to do is splice those out. There's a special class of enzymes called spliceosomes that are going to accomplish that. And so let's take a look at that. And so we want to talk about RNA splicing now. And so that's kind of like the most remarkable part of this whole process. And it involves these large proteins that come along and edit the RNA before it leaves the nucleus. And so, as I was mentioning before, these non-coding segments, or introns, can be rather large, and those need to be removed. And so that the final messenger RNA that leaves the nucleus is just exons. And so usually the exon contains just one gene, and that's going to be making a polypeptide. It's funny, the messenger RNA in a prokaryotic cell could actually contain numerous genes that are all sort of associated with the same functionality, and those are called operons. But in a eukaryotic cell, it's generally one gene for one protein. I like this diagram right here because it sort of shows what's going on here. So here's after transcription is complete, the 5' cap is placed onto it, the poly A tail is placed onto it, and so now what we have to do is just simply remove these introns. And when we splice those out, then we'll have just the coding region, or just the exons, or the areas that are essential to be expressed into a protein. And so let me sort of just back away from this and show you a little, if I can, show you a little overview of this process. It doesn't go into a lot of detail, but it's kind of a cool thing to see. So out of this vastness of DNA, you can pull out a segment here, and let's say that it's going to be transcribed into messenger RNA, and so there it is. And so this piece is going to be transcribed, but there's a lot of material that needs to be removed. So these introns literally need to be pulled out, and then this is now attached together or ligated together. So this part, this intron is removed, and then over here another intron is going to be removed right over here, and then we're good to, or maybe not, and then we're good to go at this point. And so let me show you this video while we're here. This video starts off with showing a typical bacteria. It has one chromosome. It's kind of cool. RNA polymerase, only one kind of RNA polymerase, attaches to a promoter. It starts producing the messenger RNA. Notice, since it has no nucleus, translation occurs as soon as transcription is over. So you get these proteins being produced. The ribosomes jump onto messenger RNA, and so translation occurs immediately. And so what's kind of cool is these proteins that are produced off of this messenger RNA usually have something in common with one another, and they're sort of coordinated. And so this one area right here is referred to as an operon. But things are a little bit more complicated in a eukaryotic cell. We've got the nucleus, you got the DNA, it's wrapped up in many chromosomes, and so what has to happen is RNA polymerase comes along, and yeah, it attaches to a promoter, and then it terminates, it leaves, and then you've got this primary RNA transcript, or pre-RNA. So the introns need to be removed, and then the exons need to be spliced together. A 5' methyl guanine cap, a poly A tail needs to be attached, and then it's good to go. Then you're all right. We're done. It goes into the cytoplasm. Remember, that helps it attach to the small subunit, and then we were able to produce a protein. And so there you have it. And so let's look a little bit at the detail of this. And the detail of this, I'm going to go back to our class notes, back over here, if I may, and show you what we believe is occurring. And so I mentioned to you that there's this sort of enzyme called a spliceosome that pulls the introns out. But as it turns out, that this spliceosome is rather a large molecule. And so it consists of a bunch of proteins, and it has RNA coiled around these proteins. It reminds me a lot of a ribosome, but it's not composed of rRNA. It's composed of something called small nuclear RNA, or snRNA. Sorry about the nomenclature, but it's not messenger RNA, it's not ribosomal, it's SN, small nuclear RNA, and it's wrapped around these proteins, and it's about 150 nucleotides long. And so here is this small nuclear RNA, and it's sort of wrapped around this protein. And then it's funny, like you think this is detailed, but it's not showing all the detail. There's other proteins involved that come together and form this larger complex. And this larger complex is known as a spliceosome, so this whole big purpley dude. And so what happens is one SNRNP attaches to the 5' end, and another one anneals to the 3' end. And then what happens is it pinches in, forms this sort of this little loop right here, and then the two exons are brought together and the intron is excised or removed. And I think that's kind of a cool thing. And so let's actually take a look at that. This is a pretty effective video. And so here you can see, here's the pre-messenger RNA before it's edited. Here's the blue is the good stuff. The yellow is the part we want to remove, or the non-coding area. And so how we're going to go about doing that is we're going to need to cut it out. And so we're going to, transcription produces like a perfect copy. It's like a faithful copy, if you will, with high fidelity. So there you go. You put the cap on this side, you put the poly A tail over on this side. But now we have to process using spliceosomes and remove the introns. And so here's a close-up. We've got to remove this region right in here. And so as I mentioned, the small nuclear RNA wraps around proteins, and this little dude comes up and attaches here, and then predictably another one's going to come over and attach over here, over at the 3' side. And so a different SNRNP attaches to the 3' area, and then we've got these other proteins coming. Here's these other proteins attaching, blah, blah, blah. And they form this larger complex called a spliceosome. So this little area right here is a spliceosome, right there. And so this intron is now going to be removed. And so that's cool. So this is now the mature or completed messenger RNA that's ready to leave the nucleus. So I don't know, that's kind of an interesting process to me, that it needs to be edited. Further discussion is required on this matter, I think. And so let's go back to finally, here you can see an animation of the intron being removed by the spliceosome. And the biggest cool thing about the spliceosome is not just that it removes introns, is that when this was discovered, it became clear that the fact that RNA was catalyzing something, in other words, speeding up the removal of the intron, the removal, it became clear to biologists that we needed to broaden our horizon in terms of our understanding of how enzymes work. We used to really believe for a long time that proteins were the only enzymes because they were their diversity, and they speed up chemical reactions without themselves being involved. But by the discovery of a spliceosome, or in this case, these small nuclear RNA molecules, they act as, this is kind of a cool term, ribozyme, which means that the RNA itself, because of its cool complexity, since it's single-stranded, because it can form these interesting shapes, can also be an enzyme. So ribozymes function as enzymes. So RNA can be an enzyme just like a protein can be an RNA, just like protein can be an enzyme. And so the discovery of ribozymes in spliceosomes made the whole idea obsolete that all biological catalysts are protein. I think that's kind of a neat thing. And then just to comment on this in conclusion is that by interrupting, what's the story? We could talk further about this, but what's the story with these introns? Well, perhaps what is going on is that by interrupting the genes right here, you might actually provide an ability for the cell to be able to come up with some new novelties in terms of protein design. These different domains could be created. Proteins have these modular parts, and so perhaps by separating them, you're able to increase the diversity of the protein. And then the last thing that I wanted to say is that another hypothesis about all of these introns occurring is that when you separate the gene, let me see if I can illustrate this. When you separate the genes so far away from one another, or at least the exons so far away from one another, and you interrupt them by these introns, and I'll come back to this point a little bit later if this isn't clear, is that when crossing over occurs in a diploid organism between homologous chromosomes, if you separate the genes by a lot of distance, they're more likely to cross over, which increases the diversity in gametes. And so that's one hypothesis, as well as just simply it provides sort of a word bank of possible nitrogenous bases that can become excised and transpose on. And in other words, it's sort of like a little thesaurus. You never know what you might want to use. Introns are sometimes referred to as junk, not garbage. Garbage is something that you'd want to get rid of. Junk, you keep it around, you never know. You may want to use it, you may not. It could be useful sometime in the future. It remains to be seen. And so when everything is said and done, the messenger RNA leaves the nuclear pore and it's ready to go to the ribosome to be translated. So I hope you enjoyed that RNA processing and modification discussion. Thanks for watching.